The overall goal of this procedure is to use allele-specific CRISPR-Cas9 mediated deletion to study regulation of gene transcription in mouse embryonic stem cells.
The main advantage of this technique, which uses a hybrid genome to generate and analyze monoallelic deletions, is that it eliminates the need to analyze biallelic enhancer deletions that may not be viable.
This method can help answer key questions in the functional genomics field, such as which gene does a specific enhancer regulate and how dependent is that gene on any one enhancer for its activity. Though this method can provide insight into stem cell pluripotency, it can also be applied to other systems. For example, it can help us to understand how stem cells change their fate to become neurons, blood cells, or muscle.
After designing, constructing, and confirming the sequence of the guide RNA or gRNA to be used, prepare to transfect the endotoxin-free plasmids into ES cells. Using a centrifuge, pellet 1 × 10⁶ F1 embryonic stem cells generated by crossing mus musculus 129 with mus castaneus in a 1.5 ml tube at 300 × G for 5 minutes. After the spin, discard the supernatant and add 100 microliters of the resuspension buffer supplied by the kit manufacturer. For deletion of the target DNA region, add 5 micrograms each of pCas9 GFP 5' and 3' gRNA plasmids. Using a pipette, gently mix. Avoid introducing any bubbles. Next, use the electronic pipette tip of the microporator to aspirate 100 microliters of the electroporation mix, taking care to avoid introducing air into the tip. Then, plug the pipe.